terminalization gets completed by the end of diplotin or last phase, that is dikinesis. So in dikinesis, everything what happens in prophase, nuclear membrane completely gone, spindle fibers clearly visible, this crossing over is also complete. So these two sister chromatids which are visible, they fuse. So instead of a dyad, we again start seeing a simple chromosome. So it is a reverse of this. What happened here? There was a bivalent. Two chromosomes, homologous, they came together. They underwent vertical splitting. Each chromosome formed a structure with two arms. We called it a dyad. So we saw tetrads. Then the non-sister chromatids, they exchanged the genetic material. In this case, that is in dikinesis, reverse takes place. That means after crossing over, the tetrad will again come back to this stage. Because why was this change required? This change was required for crossing over. Crossing over is complete. So now it has to come back to its original thing. So two centriole at two poles with their aster fibers and the chromosome pairs. So again we start seeing a homologous chromosome pair So there are two homologous pairs, and after this exchange of genetic material, crossing over is complete, terminalization has taken place. You know, there is a little exchanged part which we can draw here. So here these two arms, because they exchange the genetic material, they have this exchanged material. So let us write the change here. It is called terminalization. This terminalization normally gets completed by the end of diplotin. So these two are the events which are taking place in diplotin. Dikinesis, from the tetrad stage you again come back to bivalent stage, and here we start seeing the spindle fibers very clearly. Here also spindle fibers are visible, but because the chromosomes are the more important things here which we need to represent, that is why spindle fibers are lightly drawn. So now all these 5 subphases which have taken place are the part of prophase. So from here till this last one, this was prophase I of meiosis I. This is the longest phase of meiosis. Even in mitosis, meiosis, if we compare these two, this is the longest phase. Let us recap all the things which have happened here, because this is the most significant part of the division. Interphase cell with diploid number of chromosome enters the first phase of meiosis I, that is prophase I. Prophase I is subdivided into 5 subphases: leptotin, zygotin, pachytin, diplotin and dikinesis. In leptotin, that is the first one, condensation of chromatin fiber starts, but condensation takes place at intervals. So these fibers, they look beaded, as if there are beads in between, intermediate thickenings. Plus, one interesting thing is that all fibers at one end are converging to one point, and at the other end they are diverging. So it gives an appearance of a bouquet. So it is known as a bouquet stage. Further condensation of chromosome takes place in zygotin. So here we start seeing the chromosome structure, and these chromosomes, they pair up. Pair is of homologous. What are homologous chromosomes? The chromosomes which we have received from our parents. One out of the pair comes from mother, one other chromosome of the same pair comes from the father. We started with 4 chromatin fibers, so there are 2 pairs. These pairs are homologous or homologous chromosomes. They are held together. Unless and until they remain together, all these processes are not possible. So how are they held together? They are held together by synaptonemal complex. What is this complex? It is made up of thin fibers which connect these 2 chromosomes. These fibers are made up of nucleoprotein. When we enter the next phase, the cell, each chromosome of the homologous pair undergoes vertical splitting. As a result of this, we start seeing a dyad. Because 2 chromosomes are together, we would call it a tetrad. So 2 dyads together, a tetrad. The chromatids of 1 chromosome are called sister chromatids, and the chromatids of 2 chromosomes, they are called non-sister chromatids. Crossing over is possible only between non-sister chromatids. So in Pachytene, a cross-like structure, X alphabet-like structure is formed, which is called chiasma. After this chiasma formation, exchange of genetic material will take place, which is called crossing over. Crossing over, this is important, crossing over may start at the end of Pachytene or in early diplotin. So there is this overlap which is seen. The cell enters the next phase, which is diplotin. Here crossing over takes place, genetic material gets exchanged and terminalization also takes place. That means this cross which was formed, exchange of material, and then these chromosomes separate also. After they separate, in the next phase, that is dikinesis, we are showing these separated chromosomes with exchanged material. And all those other changes which are characteristics of prophase, they are also seen here. That means spindle fibers properly formed, asters properly formed, nuclear membrane completely disappeared, nucleolus gone. So everything is done. Now the cell is ready to enter metaphase. So now this cell enters the second phase of meiosis 1, which is called metaphase. In metaphase, the chromosomes, they arrange on the equatorial plane. So if these 2 are the poles, an imaginary line in the middle would be the equatorial plane. Two centrioles at 2 poles, their aster fibers and spindle fibers also. But let us make these chromosomes first. The chromosomes arrange in such a manner that the pair comes on the equatorial plane, and the pair is like this, so that one chromosome moves towards the other one pole and the other goes towards the other pole. So these chromosomes, they would arrange themselves on the equatorial plane in such a manner that one chromosome of the pair faces one pole. This is the second homologous pair. And now the spindle fibers. These are the chromosomal ones. These are the continuous ones which are not connected to any chromosome or anything. Now when they separate, these chromosomes, when they separate, when next stage, that is in anaphase, what is happening here is these 2 chromosomes were separate. Each chromosome was having its own centromere. So one chromosome with its centromere, the other chromosome with its centromere would separate. So this splitting of centromere will not take place. In mitosis, anaphase of mitosis, we said that the chromosomes, let me draw it here at one corner, we said that this is the chromosome, this is the original chromatid, and this was the copy which was formed during S phase. So when these two separate, the centromere splits from here. Whereas in this case what is happening is, there is one chromosome with its centromere and other chromosome with its centromere. So in this case there is no splitting of centromere. This is a very important difference. So now metaphase, chromosomes arrange on the equatorial plane in such a manner that one chromosome of the pair faces one pole and the other chromosome faces the other pole. Now the cell will enter, let us write metaphase I here. So now the cell enters the next phase of meiosis I. It will be called anaphase I. What happens in anaphase? These fibers, spindle fibers, they start to contract. When they contract, they pull the chromosome towards the pole. Aster rays. Because in case of these division, cell division diagrams, too many labels are not there. So we have to be very particular while drawing that each and everything should be there in the diagram. So let us use the same color. This is the blue one, this upper one going towards the upper pole. The other is the yellow one which is going towards the other one. Same thing with the other pair. And the spindle fibers. This is the continuous one, again continuous, again continuous. Between the separating chromosomes, again there are interzonal fibers. So same thing, what we saw in case of mitosis, same type of spindle fibers. These ones are chromosomal, which are connected to the centromere of the chromosome. The one which run from one pole to another pole, they are called continuous, and the fibers which appear between the two separating chromosomes are known as the interzonal. So this remains the same. Again, we are not drawing early anaphase I and a late anaphase, but in late these chromosomes will go even closer towards the pole. So this is anaphase I. This will be followed by next phase, that is telophase. Telophase is when the nuclear membrane reappears, nucleolus reappears, spindle fibers disappear, reverse of prophase. Now sometimes telophase I does not take place and immediately it goes into cytokinesis, but we will show this telophase here. And during telophase itself, the cytoplasmic constriction also is visible. That means the plasma membrane starts showing in foldings, because this will be followed by cytokinesis. Aster fibers, spindle fibers, they disappear. Chromosomes become thinner, that is, they change into chromatin again. So how many chromosomes have gone towards one pole? One blue which we were talking about here and the pink one. So these two have gone up and two have gone down. How many chromosomes were there when we started? 4. So how many are going towards one side? 2. And okay, let me use the same colors so that we are able to keep a track of it. And this yellow one has gone here and a green one. Nuclear membrane will reappear. So this would have its nuclear membrane and this would have its nuclear membrane. Chromosome number has been reduced to half. We said meiosis 1 is reductional division. So you start with 4 and you end up getting 2 daughter cells with 2 chromosomes each. This will be followed by, okay, this is Telophase I. This is followed by Cytokinesis 1. Again, Cytokinesis might not take completely, it can be an incomplete thing also, but what would happen in cytokinesis? These infolds of plasma membrane will get deeper. Both cells have their centrioles, their intact nuclear membranes, and each nucleus has 2 chromosomes. All other structures are there, but as we have not drawn them from the beginning, we are not drawing nucleolus and other things. This division would get complete after some time and we would get 2 daughter cells. These 2 daughter cells remain together because together they have to go through meiosis II. So this is Cytokinesis 1. Now both the cells, because these are daughter cells, they are smaller in size, both of them together will go through interkinesis. Interkinesis is where they would accumulate protein, ATP and the material for the next division. No S phase, that means DNA doubling will not take place. Now before we take up meiosis II, let us talk about one more very important thing which we know but we have not shown in the diagram. What is that? That thing is that the cell which we started with, that is interphase cell, it has undergone G1, S and G2. G1 and G2 is for growth, accumulation of things. What has happened in S? In S, these chromosomes, they have made their copies. That means each chromosome is with a copy. DNA has been doubled. So we are writing it as 2n. But what is the genetic material like? It is equivalent to 4n. That is what has happened in S phase. We have not shown it. But now as we have done this, I am just going to modify this. That means here is a copy. We purposefully did not draw it. Reason? It would have become very complicated. So we know that with each chromosome there is a copy. So when these 2 have separated, this has gone with its copy, this also has gone with its copy. That means each chromosome is going with 1 copy. But why are we counting it as 2 chromosomes? Because the number of chromosomes is 2. Genetic material, DNA is double and this would get separated in meiosis II. So we have to remember very important thing that in the beginning itself, all chromosomes have double the DNA content and they are keeping them with themselves. So now when these 2 daughter cells are formed, each daughter cell has half the number of chromosome, 2 in these cases, and each chromosome has a copy which was formed in interphase when the cell entered the cell division. Now let us start with meiosis II. These 2 daughter cells will enter meiosis II together. So for our reference, let us draw the 2 daughter cells, because these 2 daughter cells are going to enter meiosis II. And as we said, these 2 cells will enter into the next phase of division together. They are together. That means they remain attached. Each cell is having its centriole, each cell is having its nuclear membrane, and each cell is having 2 chromatin fibers. These are 2 daughter cells which are formed after meiosis I. So, the 2 cells which we got after meiosis I are these. They have half the number of chromosomes. So, these 2 daughter cells are haploid, half the number. Original parent cell had 4 chromatins, here each one is having 2. But now we will say that each chromosome has a copy. When was this copy formed? This copy was formed in interphase. These cells undergo interkinesis. Interkinesis. Interkinesis is a period of growth which is like G1 and G2, but here there is no S phase. So, all that DNA replication which is required has already taken place in interphase. Now, these 2 cells undergo this short, very short growth period just to accumulate those proteins, ATPs and other molecules which are required for the further divisions. Now, these cells enter meiosis II. So, from here starts meiosis II. First phase of meiosis II is Prophase II. Again karyokinesis, nucleus is going to divide. Let us keep in mind that it is similar to prophase of mitosis. And again, we can draw early prophase and late prophase, but now as we have done it once, we know what all changes are going to take place. Where is the centriole? At this pole and this pole. Why at these poles? Because for the parent cell, these were the 2 poles. Now, the centriole of one cell and centriole of the other cell, they split and they move towards the poles. In this case, the poles are not going to be these 2. The poles are going to be at right angles to the first axis. First axis was vertical, so in the next case, right angles, that means horizontal would become the pole. So now the centriole splits and one centriole, if we draw 2, one goes towards this side and other goes towards this pole. So after this, the 2 cells will appear to have one centriole here, other centriole at the other pole. So the orientation of poles has changed. In the first division, it was vertical axis of the pole. In the second case, it's going to be right angles to the first. So here it is horizontal. But if it was horizontal in the first case, it would have become vertical in the second case. So it is right angles to the first, whatever is the first axis. So this is again prophase. The nuclear membrane starts to disappear. Here it is completely gone. So we can say this is early and this is late. Prophase II. Chromatin fiber starts to condense. So we start seeing chromosome-like structures. Each chromosome has a copy, and this copy was formed during interphase. This thing we have to sort of fix in our heads, that each chromosome coming here is with a copy of the DNA. So this is having double DNA. By the end of prophase, we start seeing spindle fibers, and see the orientation axis is all right angles to the first case. In case of the first division, meiosis I, the spindle fibers look vertical. Here they are horizontal. And these chromosomes, because the nuclear membrane is gone, these chromosomes, they are in the cytoplasm, open in the cytoplasm, or scattered in the cytoplasm, with their copies. And these copies, or the chromatids of these, they are attached at the centromere. So the centromere is common, they are fused at the centromere. And when these two separate, centromere will split. So centromere splitting is seen in mitosis as well as meiosis II, and that is why meiosis II is considered same as mitosis. This will enter metaphase II. All changes must be drawn in both the cells, because both the cells have to complete the division. Till they complete, the division will not be resulting into 4 haploid cells. So these two cells again, the chromosomes in metaphase arrange themselves on the equatorial plane. So if these are the poles, the equatorial plane is an imaginary line in the middle. So this would be equatorial plane for one cell and other would be here. Again right angles to the first case. Chromosomes appear or arrange on the equatorial plane. Each chromosome is with the copy. So let us draw the copy here. And they are fused at the centromeres. Spindle fibers, they have already appeared by the end of prophase. Some fibers will attach to the chromosomes. These are called chromosomal fibers. Some fibers are going to be continuous, that means not attached to the chromosomes. Same in the other cell also. These chromosomes are on the equatorial plane of these cells. This is metaphase. Now these chromosomes will separate. Separation will take place in anaphase, Anaphase II. It is a part of meiosis II. So these two cells will show contractions of these spindle fibers pulling the chromosomes apart. Which chromosomes are getting pulled apart? If you are talking about this chromosome, the original chromosome and the copy which it made, so one will go here and the other will go here. Are they fused? They have the same centromere. So here centromere is again going to split. So the centrioles at two poles, spindle fibers, this is the continuous one, the chromosome after the centromere undergoes splitting, one goes here and the copy goes towards the other pole. In this case, again same thing, one chromosome after splitting of centromere goes towards one pole and the copy goes towards the other pole, and between these two separating chromosomes would appear interzonal fibers. Same thing will be seen in case of the second cell, second daughter cell. So, one chromosome towards one pole and the copies towards the other pole, and interzonal fibers. Early anaphase, the chromosomes have just separated. Towards the late anaphase, they would reach towards the poles. Next phase would be telophase. The two cells are still together. Their membranes. What has happened by the telophase stage? Telophase, as we know, is the reverse of prophase. So, nuclear membrane will reappear, chromosomes will thin out into chromatin fibers. So, this is the centriole. These two chromosomes would reach towards one pole. So, let us draw these here, and they will thin out. That means chromosome will become thinner and will change into chromatin fibers. Two would reach the other pole, same with the other cell also, other daughter cell, and here same nuclear membrane reappears. So, now this would have its nuclear membrane and this would have its own nuclear membrane. During mitosis and even cytokinesis 1, we said by the end of telophase, cytoplasmic constrictions start. The plasma membrane starts showing constrictions. The constrictions appear on the equatorial plane. So, we will start seeing a constriction in the plasma membrane here, equator of this cell, equatorial plane of this cell, and here. This is telophase. Telophase II. After this, it will enter cytokinesis. In cytokinesis, this cytoplasmic constriction will become deeper. This side constriction will get deeper. The other cell, its cytoplasmic constriction. Each cell is having its own centriole. Their own nuclei, complete nuclear membranes. Each nucleus is with two chromatin fibers. By the end of cytokinesis, this is cytokinesis 2. By the end of this, we would have 4 daughter cells. 1, 2, 4 daughter cells completely separated with their individual nuclear membranes, centrioles, and each one with 2 chromatin fibers. The cell with which we started was a diploid cell with half the number of chromosomes. So, this was the cell, it was diploid. This cell underwent meiosis 1. As a result of meiosis 1, what happened? We got 2 daughter cells, each having half, okay, let us put the number as 4 here because we started with 4 chromatin fibers. So, After meiosis 1 we got 2 daughter cells, each one with 2 chromatin fibers. Both these cells, they underwent meiosis 2 and we got 2 cells from each having 2 chromosomes. Why was this 2 coming here? Because when we started with the cell had 4 N copies. When meiosis 1 took place, these 2 chromosomes got separated, but each one went with its copy. And after meiosis 2, the chromosomes and their copies got separated. So where is doubling of DNA taking place? Interphase only, at the beginning, not during interkinesis. So as a result of meiosis, we get 4 haploid cells. These cells are half cells or gametes. So these can be sperms or eggs. So gametes are produced as a result of meiosis. So these 4 cells which we have are 4 daughter cells. Reduction takes place in meiosis 1. So meiosis 1 is reductional division. And what is happening in case of meiosis 2? In case of meiosis 1, parent cell gave rise to 2 daughter cells with half the number. Here these 2 cells will give rise to these 2. So the chromosome number of the new daughter cells is similar to their parent cells. So this meiosis 2 is equational. Equational division. This completes meiosis. So prophase 1 is the most complicated one, other things remain the same. Go through each and every step, important points, remember, because the questions which are asked are based on what happens in this particular stage, or the stage is given like bouquet stage, is which stage, when do we start seeing chiasma, when is synaptonemal complex formed. So we have to be very particular about each and every stage and the changes which are taking place in all these stages. So meiosis is complete now. Let us see the types of meiosis and then we will compare mitosis and meiosis.